Hello, everyone. I'll be presenting the effects of different membrane scaffold proteins on GPCR functionality in lipid nanodiscs. So our lab studies GPCRs, G protein coupled receptor. Why do we study GPCR? GPCRs are very important drug targets. More than 30% of all drugs approved by the FDA target G protein receptors, and a lot more G protein receptors are not yet studied. It includes many functions relating to major human systems, as shown in this figure. To study GPCRs, we use a mimetic system called nanodisc. These nanodiscs mimics the human cellular membrane. Yes.
I'm not seeing, I might be the only one. I'm seeing conclusions and discussion. Okay, there it is. Thank you. Now I'm seeing
Sorry.
it. That's all right.
Okay. Can everyone see the introduction? Yes. Okay. So we use nanodiscs as a tool to study GPCRs by introducing membrane protein in this molecular sushi per se. The membrane scaffold protein will serve as a molecular belt that encloses the synthetic phospholipid bilayer that will include compositions that are closer to the native human cellular membrane. We can introduce anionic lipids as well as cholesterols. And so what I've been working on this summer is producing these variant membrane scaffold proteins through mutations, through polymerase chain reaction, PCRs, and transforming them into E. coli BL21D3 cells and expressing them in TB media, following very typical protein production protocols. This SCS page gel figure shows successful expression of MSP1D1 as well as the successful purification of such protein. And lastly, we assemble nanodiscs with these membrane scaffold protein with the labeled A2AR adenosine receptor, as well as introducing the different phospholipids. So the two phospholipids that we use is this retionic POPC and the anionic POPS, following very typical compositions in human cellular membranes. So the results, we use 19F NMR to see the different conformational states in the particular membrane receptor. A2AR has a very unique engineering where we can attach TET to the A2AR variant A2A9C through thiochemistry. These produces a 19F NMR with very resolute peaks that we can correspond to active states of the membrane receptor conformation states. So some of the results we can see in the regular MSP1D1 nanodisc with a phospholipid composition of 78 molar percent POPC and 22% POPS, we see the abundance of P4 active states. However, when we move on using the MSP1E3D1 nanodisc, which is a larger size for the larger size variation of the nanodisc, we no longer see the P4 active states. This shows that the conformational dynamics of GPCRs are heavily influenced by this protein-lipid interactions within the native cellular environment. We can induce these differences by introducing variations within the membrane scaffold protein. Some of the concurrent works that we've done is introducing substitutions within the native MSP1D1 from replacing normally uncharged residuals to charged residuals in hopes to introduce artificial lipid asymmetry within the lipid distribution of the nanodisc. Through this work, we hope to improve nanodisc design for future drug research in order to more accurately simulate a human cellular membrane environment. Further research that are planned are using complex formations with the G protein as these receptors are normally coupled in a complex formation with G protein to study more accurate functionality such as in the regular human cellular membrane. Thank you for listening. Thank you for my mentor, Dr. Matthew Eddy, as well as his graduate student, Viraj, in helping me through the summer. And thank you all for listening.